Welcome to the NIHR Dementia Researcher podcast, brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK and Alzheimer's Society, supporting early career dementia researchers across the world.
Hello, my name is Dr. Fiona McLean and I am an Alzheimer's Research Fellow at the University of Dundee, where I investigate how metabolic diseases like obesity and type 2 diabetes can lead to diminished brain function. And within that area of research, I have a specific focus on the blood-brain barrier. Today, I'm really pleased to be hosting this Dementia Researcher episode on Parkinson's disease. With me is Dr. Sarah Marzi from the Dementia Research Institute at Imperial College London and Dr. Dayne Beccano-Kelly from the Dementia Research Institute at Cardiff University. Hello to you both.
Hello.
Hello. So now I've done a quick introduction, let's find out a little bit more about each of our researchers. So why don't we start with Dayne?
Okay, so my name's Dayne Beccano-Kelly. I'm a UKRI Future Leader Fellow and Group Leader at the Dementia Research Institute at Cardiff University. And my research focuses on Parkinson's disease and the temporality of Parkinson's disease, really trying to understand the order in which phenotypes appear in an effort to really increase the efficaciousness of therapeutics and the design of them so that we can really temporally lock in disease-modifying therapeutics.
Fantastic. And Sarah, what about yourself?
So I'm an Edmund and Lily Saffer Research Fellow and UK DRI Emerging Leader at Imperial College. And my research focuses on gene regulation and epigenetics in neurodegenerative diseases, specifically Parkinson's and Alzheimer's disease. And the main hypothesis that really underlies all of my work is that I want to understand how non-coding genetic risk and environmental risk factors act on disease risk. So what does it do to the cells that are involved, to the cells that are vulnerable to the disease, and how do these cells change their regulatory programs in response to these external and internal insults, and how might that promote the disease?
Amazing. Both sound absolutely fascinating research, and we'll get into it a little bit more in depth in a minute. But I think for our listeners, it's a good place to start is actually talking about what Parkinson's disease is and what are the classical symptoms and how does it sort of differentiate from other neurodegenerative diseases. So I'll let Dayne start with this one. So what is Parkinson's?
So Parkinson's disease is the fastest growing neurodegenerative disorder in the world. I think it's predicted actually by 2030 to surpass and exceed Alzheimer's disease in its prevalence worldwide. Wow.
And I don't think that's something many people would actually be aware of. Is actually this growing increase.
It's this growing increase. All neurodegenerative disorders are impactful. They're all incredibly difficult for people that are witnessing it happen to loved ones and obviously going through it themselves. But when you hear of one on the rise, it's obviously something that helps us all to focus and redouble our efforts in trying to battle against it. So
absolutely.
Yeah.
So what are kind of like the classical symptoms of Parkinson's?
So Parkinson's disease is typically thought of as a motor disorder. You normally end up with lots of issues with goal-directed coordinated movement. So, reaching for something to move, to a drive to get yourself a cup of tea and move it back to yourself and drink it. These sort of goal-orientated movements are disrupted by this hallmark motor tremor that you get. So you get this resting tremor, which is the sort of shaking of sort of ambulatory systems, and so you're kind of constantly shaking in this guise. And so you also though have maybe things that are less well known, things like a truncal rigidity, stooped posture, sort of masked facial expression, and so you have a lot of different things that seem to almost be counter possibly to the continual dyskinesia, the movement, that resting tremor. You have lots of rigidity in various areas of the body. but yeah, it's got a lot of disruptive motor symptoms, but there are also other symptoms, things like anosmia, loss of smell that happens quite early in Parkinson's, constipation, and these are things that maybe get talked about a lot less, but are probably just as important. And there are indeed cognitive dysfunctions as well. So.
Well, that's what I was sort of really interested about when I sort of looked into Parkinson's a bit more, is that typically we do think of it as a motor disease, and that resting tremor, which a lot of people maybe seen in people they know or even out in public, this sort of continual tremor that people typically display in their hands, I guess, is where you probably most notice it. But actually, when I was looking into it, I found a stat that said about quarters of patients actually go on and develop some form of dementia or cognitive sort of disturbances. So is that something, do you think, in the field has been sort of enough awareness, and is there enough research into the sort of more cognitive side of the symptoms of Parkinson's? Sarah, what do you think?
Yeah, so I think that is something that we're becoming increasingly aware of, and that more and more research is targeted towards. I have several colleagues who work on this now, but like you said, I think historically maybe it's been sort of ignored, and Parkinson's has been thought of as just one simple disease, but then you get these differing trajectories. I thought it was almost half of people who develop some sort of cognitive impairments at some point in time, and it might take a while, so it's usually a later stage symptom than the motor symptoms. And we don't really understand why some do and others don't, and it's really important to figure that out. Are there basic underlying differences in these diseases, or is it just a temporality? Is it a downstream effect of the original degeneration that's happening? So Parkinson's disease neuropathologically in the brain, what we see is the loss of dopaminergic neurons in the substantia nigra. So this is one very small part of the midbrain. And although it's not been too definitively answered whether none of the other surrounding dopaminergic neurons or related neuron types are affected, it seems to be quite specific to that area and to the dopaminergic neurons. And then as they die, they also stop sending signals out to the neurons that they connect to in the more in the areas at the front of your brain. And so it's thought that maybe because they don't receive signals anymore, that these neurons then also start to degenerate. But I don't think we understand it really well. I mean, I'm sure Dayne has some thoughts on how that might work with the nigrostriatal to frontal signaling.
Because a lot of your research is electrophysiology based, isn't it? Dayne, and your research kind of focuses on the early stages of Parkinson's, and you look at synaptic health. So I think that's quite a good place to start. Can you tell us about synaptic health and how it's affected in Parkinson's disease?
Yeah, sure. So following from what Sarah was saying, I'm hugely biased, by the way, because I'm an electrophysiologist. So
we're all biased about our research.
Yeah, but I'm definitely right. No, I'm obviously joking. No.
So electrophysiology is the best.
Of course. We're looking at neurons, but neurons are the thing that is, susceptible in Parkinson's disease to a degree. There are other cell types that are destroyed in Parkinson's disease. You have the gut's affected. For instance, you've got actually liver might be affected as well.
Which is very interesting about the gut. Maybe we should start with synaptic health, but we'll come back to the gut because the gut technically has some neurons in it, right?
Oh, yeah. No, so definitely. And it's,
let's not...
We can come back to that, I suppose. But if we are going to sidle back to the neurons in the brain, the main role that they have is this, is this neurotransmission, this like amazing ability for them to encode information in a temporal sense using chemical release and electrical transduction of information. And it's their specialized role. And so if we're looking at a neurodegenerative process, we probably should be looking at their normal function and how that's disrupted. And it's my belief that it's likely that this is allowing some level of susceptibility in Parkinson's. Because of this specialized role and because of the specific destruction of neurons. It must be something to do with their specific role. And so we focus on looking at temporality of the disease, looking at the earlier stages, but we think that this will be in and around functions that specifically contribute to this synaptic function, this synaptic role. So we're looking at multiple different features, calcium release that drives and helps neurotransmission release. We're looking at things like ATP production, which again modulates and affects synaptic function, and many other functions in and around electrophysiological function and dysfunction, I should say, because what we really want to know about is the dysfunction that precedes that neurodegeneration, because what we're interested in is maybe helping to modify and delay the onset of the cell death that will then occur after that dysfunction. And there's a lot of work that's gone on in the field looking into synaptic function now in Parkinson's disease. It's a lot of work that's happened fairly recently in the grand scheme of the literature.
Which is exciting.
Yeah, no, it's
fantastic. Because it means that you're right at the forefront of it. I have a question. Here's my bias, research bias coming in, because I'm obviously interested in metabolism and energy, and you mentioned ATP at synapses. So do you think that there is a role of sort of mitochondria at the synapses that's perhaps becoming defective? Do you think it could be actually something to do with sort of energy regulation at those synapses that then prevents them sort of doing their job and communicating with? I
do. Mitochondria are very well defined, that mitochondria contribute to the mechanisms of cell death in Parkinson's disease. We know that from genetic models of Parkinson's disease. Recessive models have shown quite a lot of impact on mitochondrial function, but that cell death that it seems to be contributing towards also might be, if we look earlier, there might be dysfunction of the mitochondria, and mitochondria are known to be functionally as active at the synapse. We actually just had a guest speaker up here who's really looking at that as well, Mike Devine at the Crick Institute, and it's fantastic work where he's looking at migration of mitochondria and movement to the synapses, and it's definitely involved. Myself have found some data suggesting mitochondrial deficit in their functionality can then contribute to a lack of efficaciousness of the synapse, so they don't work as well when they have a decline in calcium input into the mitochondria, which will decrease their ATP output. And so
makes
they're
sense,
all
right?
linked. It does definitely make sense.
Because when you think about just as people and existing, like, we all need energy to do our jobs.
Exactly.
So it makes absolute sense that on a cellular level, these cells need energies.
Something that's so energy demanding as synaptic
right?
information, transmission of electrical signal in dopaminergic neurons, they're huge. They have these long unmyelinated projections that extend for extended period of time. And they fire at this pacemaking activity.
Because they're unmyelinated, does that mean that they sort of need more energy because they don't have that insulated?
Likely, it's likely this, and also the fact that they fire. This is sort of almost like a heartbeat, a regular pacemaking. So they're continually active. It's not. So this requires a lot of energy to drive them. So maybe just to quickly go back to what Sarah was saying, the interconnection between all of the neurons in the brain. We're talking about these dopaminergic neurons, specifically the ones in the substantia nigra pars compacta versus the reticulata. The ones in the substantia nigra pars compacta are projecting up into the striatum and the basal ganglia, which is just a mess of crazy connections. It's like a really complicated area of the brain. But if you can imagine that they're all dependent and reliant upon each other, you've got GABAergic neurons reliant upon cortical neurons. So one disruption to one area might then manifest in a different area. So actually that's what some of the things that we're working on is it might not actually be starting in the dopaminergic neurons. It might well be starting elsewhere, and dopaminergic neurons are just the ones that are most susceptible to said change and kind of die first.
So do you think that area of the brain could be maybe thought of as like the London tube map? So it's kind of like, it's all, cool. There's loads of different paths and loads of different, it looks really crazy and complicated, but actually there is an organisation to it. But if you get a problem on one line, it can affect other lines, even if, they're meant to be different things. Is that quite a good analogy? Or
it is. I'm sure Sarah can talk about the disruptions on the tube lines and how everybody then rushes to the district line and you can't get across. It's just a nightmare. But no, I think that's actually a really good analogy. It's an organic development. I'm definitely going to use that. That's amazing. I'll bring up the tube map and then I can talk about all.
It's not like the Glasgow tube map, which is like a circle.
Yeah, nice and simple.
A little bit more easy to understand. So, Sarah, your research investigates Parkinson's disease from a different angle to Dayne's, and you study something called epigenetics. So, can you just explain in a little bit more depth what epigenetics is and why is it relevant to Parkinson's disease?
Right. Okay. So, epigenetics is basically all the mechanisms that our body has, biochemical mechanisms, to regulate gene expression. Because every cell in our body has DNA, and it has exactly the same DNA. In fact, it's your personal DNA code that encodes all your information that you've inherited from both of your parents. But then how does every cell know what to do? Because our body is made up of hundreds of different types of highly specialized cells that do very different things. You might have a muscle cell that's involved in muscle activation and movement. You have skin cells that cover your whole body. You have receptor cells in your eyes, and then you have lots of different cells in your brains, like the dopaminergic neurons that degenerate in Parkinson's. And they look very different in terms of their shape and morphology. They do very different things, like neurons would be sending electrical signals, for example, but not all the cells in our bodies do that. So how do they know how to become what they are? That's epigenetics. So epigenetics is basically, you can think of it as an annotation that tells this really, really large genome, your DNA, which parts of it to use. So like, silence down all of these genes and then use this class of genes because you're going to need it to for your calcium signaling, whatever receptors, produce all of those proteins because you're going to need it as a neuron. And so the one really important part where that is seen most clearly is cell development and differentiation, and that is thought to be driven primarily through epigenetic mechanisms. But then it's not completely locked in and stable for the rest of your life. It's also a bit dynamic, and it can respond to environmental factors. So, for example, when we have an immune activation, when our body is infected by something, immune cells will react to that. So our body needs responsive mechanisms that can react to things that come in from the outside or also from the inside, or regulate temporality throughout the day, throughout the life, throughout different cycles. And so, therefore, epigenetics, these mechanisms allow some sort of temporal dynamicness and flexibility. I study these mechanisms pretty much to interpret how environmental risk and how genetic risk will act on the disease. And we know that both environmental risk factors, but also variation in our genome, can change the epigenome in specific places, in specific cell types. So really taking a deep dive into that and understanding what cells are affected by these given changes, where is it changing, what is it doing to these cells. So I did want to come back once more to, because you were talking about it, the mitochondria, which also from my angle onto the disease have become really prominent, because both when we look at the genetic risk factors for Parkinson's, sporadic, but also more familial Parkinson's disease, there is certainly a class of genetic variants and mutations that we know specifically affects the mitochondria.
That's so interesting.
Yeah, and equally from the environmental side, one of the best known and strongest associations for environmental risk factors are actually pesticides, so agricultural pesticides. And one really well-known one is called rotenone, and it's primarily a complex 1 inhibitor. So it inhibits the function of complex 1 respiration in the mitochondria, and therefore, energy processing and energy delivery through the mitochondria. And it results in a substantially increased risk of Parkinson's in humans, and also administering this to animals, it's done in rats, for example, will give the rat a pretty much Parkinsonian symptom. So the rat will get the tremor, will get the rigidity, and I think eventually if you do it long enough, also you can test some of the cognitive symptoms as well, and you can see some of the cellular dysfunctions that you would see in the human brain as well in the rat. So it seems to represent Parkinson's as a syndrome in the animal really well, and it causes the disease, so to say, both in the human and in the model, making it a really convincing and strong risk factor, and it acts primarily on the mitochondria. So there definitely is a really strong link there.
actually have heard of rotenone, and I use it in my own experiments because I do metabolic stuff, and I use it to shut down the mitochondria. And I actually didn't know that it had previously been used as a pesticide, and now I know what I use it for in the lab. That's terrifying.
Yeah.
Oh my goodness.
Yeah, it is,
that
right?
should not be used as a pesticide.
You say previously. So it has previously been used in places like the European Union and the US where it's forbidden now, but it is actually still used in several other countries, I think in Brazil and in India, for example. And so it's really important that this association is well known, so I think there needs to be more policy being to ensure that these kinds of toxic substances kind of get regulated and phased out across different places.
It's not the only one, right? So there's quite a few pesticides that have been linked. There's work done by Biarritz and other people over in the States, and then they found that there's a number of different pesticides that could contribute to the decline and maybe Parkinsonian features, and then our differences in our own genetics may increase the risk of that by not being able to break it down as well, or breaking it down faster. And it's interesting because some of those have been presented to various governing bodies, and some of those have been removed from being used and then sometimes been reintroduced despite the information being present. So that's a worrying
Not good. It is actually interesting because I've just been thinking while you were talking, and my grandfather had Parkinson's disease and he was a farmer. And now I'm sitting here thinking,
did he ever?
I wonder what
he ever
driving?
Yeah, I mean, he was in his 90s when he died, but he had Parkinson's for a while, and he was a farmer for a long time. So yeah, it does really make you think. And I'm sure there's been studies looking at farmers, especially we have such a big agricultural in the UK. So
association actually comes from studies of farm workers who have extremely direct and constant and continuous exposure to these toxic substances.
Wow.
They have also done some stuff where they've looked at the drift to like things like nearby schools and seen the amount of pesticides that are in the walls. They can tell how much has been used nearby, and the rate of Parkinson's in those towns, usually in the States, but in those towns, because when you see those films where they fly the planes over and they crop dust, basically.
Yes.
Obviously, that's going to drift on the wind, and so it's really bad for any neighbouring places as well.
And of course, this is then going into our, into the water. It's going into our ecosystems, and it's probably doing a lot of damage that we don't even know about. Because if we only know about the association with Parkinson's in humans, goodness knows what it's doing to other species as well. So, oh my goodness, quite scary. So,
pesticides is just one class. There are lots of other things that have been
exactly.
Well, that was my next question. Yeah,
then we wonder why it's on the rise.
Well, yes. My next question was aside from pesticides, what other kind of environmental and genetic factors are there? And do you know what the split is? Like, how important is genetics versus environment?
This is my kind of question.
Go for it.
Usually we would estimate something like that, how genetic is a disease, by looking at twins. So you would look at twins that are genetically identical and then twins that are dizygotic, so that are like siblings, essentially, sharing 50% of their variants. And so then you compare how often does it co-occur in one versus the other, and then you could quantify how heritable it is. So Parkinson's, I would say, is really only moderately heritable. It's around 20 to 40% across different studies that have estimated it. Whereas, for example, in contrast to Alzheimer's disease, which we think is around 70%, so that's quite a bit higher. That's not to say that the genetics isn't important, and I would say the genetics and the genetic studies in Parkinson's have been really thorough and really good and have shown us lots of potential pathways and mechanisms into the disease. But that's just to bear in mind that environmental factors are going to play a more important role in this disease than in others, and that we also need to look at things like gene-environment interactions, like Dayne was saying. So maybe an environmental exposure affecting different people differently depending on their genetic makeup.
Yes, absolutely.
So
On you go,
Sorry,
Dayne.
go over. That's fine.
No,
So,
I was just going to say that's really interesting. And
it is, isn't it?
please
I think
jump
there's,
in.
for the longest, just to add on, the longest time, people thought Parkinson's disease was just environmental because of that reason that Sarah's just said, because it's not as a prominent as a feature as in other disorders, neurological disorders, until the advent of, or the finding of alpha-synuclein mutations, which causes slightly earlier onset Parkinson's disease. But
So
there's
just
a number
for
of them that
our
we find.
listeners,
Yeah.
could you explain about the links between alpha-synuclein? Oh my goodness, can't speak.
Alpha-synuclein.
Parkinson's.
Alpha-synuclein is a no. Alpha-synuclein is a very small protein, synaptic protein. It's involved in membrane curvature, and think that it's very important in neurotransmission and other functions like this. In Parkinson's disease, it starts to aggregate, and you start getting these intracellular inclusions known as Levy bodies, which after Fritz Levy, which basically are one of the main ways that you can identify Parkinson's disease post-mortem, because you can only define definitively Parkinson's disease post-mortem. So you're looking for these kind of inclusions. And so when you actually have, you can have things like point mutations in alpha-synuclein, which causes or seems to increase this level of aggregation, and synuclein triplications, which is one of the earliest ones to be discovered. So you've got triplication of alpha-synuclein gene, and then you've basically got increased levels of it, which theoretically and actually empirically leads to an increase in the aggregation. It does cause earlier onset. We'd just like to point that out. And then actually in those mutations, you probably get more fulminant pathology that's probably closer to MSA. And I think we were talking about it just before, but Parkinson's disease is a bit of a spectrum, actually, if we're actually looking at it objectively. And it's to say that, it's just one thing. It's a bit hard to sort of define it like that, because the synuclein is definitely involved at some stage of the disorder. But when you have these mutations, it causes earlier onset and these differences in pathology. Whereas if you have something like LRRK2, which is more later onset, it's more similar to the age of onset of sporadic Parkinson's disease, you basically have less of that pathology and more cognitive impact. And so it's good to look at all of these. I think all of the models are really useful. They all provide us with different information. I think they definitely, you have your autosomal dominant forms, which cause later onset, which are more similar to the sporadic forms. You have your recessive forms, which cause earlier onset. And then you have mutations that in the genes that cause risk factors like GBA, glucocerebrosidase, which is an enzyme found in lysosomes, which helps the processing of lipids. And mutations in that increase a person's risk factor, but the penetrance isn't as high. And so that 20 to 40% is probably a bandwidth of how much penetration there is for how much penetrance, there is for the genetic factors. And
that's interesting. So I guess maybe if there's a spectrum at some point, hopefully in the future, if you get a Parkinson's diagnosis, it'll be maybe more tailored to you have this subtype, and then that can help towards sort of more targeted treatments and precision medicine, which would hopefully give people more successful treatments.
So I don't know. Should we discuss it? Let's all sit
go
down.
for it.
No, but unless of course if we find commonalities, like a common thread between all of them,
right?
But then that is accelerated in some and lengthened in the other. So if we found one common theme, sort of trim out the fat, then perhaps we can use it. Just that modifying that one common theme in all of them. I mean, that might be a longer shot. But there is something that's similar.
It's two approaches, right? You either try and find something that is affected in everything and hope that it improves it all, or I guess, if that doesn't work, then you start to look more in depth at these sort of different subtypes.
Yeah, it's just the rate it might be affected. Yeah, so it could be that.
I mean, that would be wonderful to
look at all the models.
To find the superpower drug that cures them all.
Oh,
but, realistic.
I know, that would be amazing.
I'm a bit pessimistic, maybe not realistic, pragmatic, and I think maybe as we've seen in other complicated diseases like cancers, maybe there are subtypes that do distinguish themselves through certain biological pathways and mechanisms that make them easier to act upon, and then at least we can target those.
No, I do agree. Just pragmatic, arguing devil's advocate. There's possibilities, I suppose, in both ways. I'm not married to the idea.
You've got to look at it all, though.
Yeah.
Because if you don't look for it, then you'll never find it.
Yeah.
So I guess talking about risk factors, is there anything that we can do as individuals to lower our risk factor for Parkinson's disease?
Take it, Sarah.
You have to live in the right areas. Probably don't live where the pesticides are. We didn't quite.
Secluded islands.
Yes,
somewhere.
we didn't quite touch upon what the other big classes of environmental risk factors are.
Let's talk about that.
So, other big classes would include heavy metals. So people who work in mines of different sorts, copper mines, mining heavy metals, have a substantially increased risk of developing the disease. Then we've seen pretty good evidence for industrial solvents like halogenated chlorines, basically like tetrachloroethylene and other variations of those that are quite strongly associated again with Parkinson's. And we've seen this especially in people who have dramatic exposure, so who literally work on top of a vat with that industrial solvent decreasing, I don't know, metal components in automobiles or whatever. But we've also seen it in some contaminations of sites. Again, in the US, there's this famous military base that got a big spillage into the groundwater of tetrachloroethylene. And then after many, many years, because it takes a lot of time and these exposures build up, you see an increase in Parkinson's occurrence. So those are two big classes. And once they're in the environment, some of these are really hard to break down as well. Like the tetrachloroethylene, TCE, unfortunately forms structures that are relatively stable for potentially decades. So it is a problem.
My
but
goodness,
so we need to, I guess that's more at the policy level, ensure that politics knows about these risk factors and acts upon it and minimizes the usage and thereby the exposures. Some other things are a bit more lifestyle factors. So one really puzzling one is that smoking is actually negatively associated with Parkinson's disease risk.
That's so weird.
Which is weird.
Right, we're not advising that people take up
Disclaimer.
Disclaimer, we're not advising you take up smoking.
Other really terrible diseases like lung cancer and cardiovascular
And dementia in general, actually, because
so
I was going to say, is it not the reverse in something like Alzheimer's disease?
That's
But
right. But it's also not known in entirety whether that is a causal association, because as a devil's advocate, one could argue that maybe the general prevalence of dopaminergic neurons, so how many you have to begin with as a human, might be associated both with your propensity to smoke and Parkinson's. So saying that if you have a lot of dopaminergic neurons, because dopamine is actually involved in the reward circuitry in our brain. So if we do something that we like, our brain goes like, oh, we're happy about this. And that might be things like smoking or drinking alcohol. And
effectively,
having more dopamine activity might predispose you to become addicted to things more easily. So that could also be an underlying cause. That has not been
fascinating.
answered.
how risk averse you are and how sort of risk taking you are, it could be affected by dopamine.
And what about stuff like exercise and diet? Sorry, back to my bias point of view from my research.
I think that's fair, actually. I think exercise has been shown to be very effective at maintaining your level in Parkinson's, for instance, and even perhaps staving off deterioration.
So slowing kind of progression.
Slowing the progression, it's been shown that can be enormously helpful. And so for the similar reasons, exercise and eating well is never a bad thing, right? So it's got to be something that we could utilize in our daily lives to improve upon this. It's just that when we've seen during Parkinson's that you utilize an increased level of exercise and coordination and control, things like dance, things like boxing. I think I was watching the same documentary that happened on on ITV the other day with Jeremy Paxman, and he was talking about how people are enjoying things like bowls and anything that can needs coordinated, controlled movement. But it's there's all sorts of information that seems to be tied with things like increased blood flow, increased concentration. And diet has been also linked. Obviously, we've talked about the gut earlier, and there's the idea that it's a sort of a bottom-up progression of the disorder. You can see sort of Lewy body pathology and the spread of dysfunction and destruction up from the gut and then into the brain. So
could you explain that a bit more? Because I find that fascinating. So what is there early changes seen in the gut? Is it linked to gut microbiota? Is it linked to sort of signaling from the gut? Like, what is this connection?
So I've got to say that the gut microbiota for me is not my strength. However, I know that that there is a lot of work going into looking into that. There is definitely early signs of pathology within the gut, and you also have the enteric neurons going up from the gut that are affected in Parkinson's disease. It seemingly occurs incredibly early, and obviously we were talking earlier as well about constipation being one of the main features, which actually occurs pretty early. And I would actually argue that there is a lot of the non-motor features that are affected pretty early in Parkinson's. It's just that the motor features are prominent. That's usually what people come to the clinic for. But you have all sorts of issues with neuropathic pain or anxiety, chronic anxiety, or sleep behavior disorders. Getting to sleep, remaining asleep, and having a restful sleep is completely disrupted in Parkinson's. And sorry, not to digress too much, moved from sort of one non-motor feature to the others, but that does turn up very early sometimes, well, quite often earlier than the motor features, and sort of progress with time so that they become more prominent later, but they are triggered earlier. So you can have people with REM sleep behavior disorder that convert at quite a high rate to Parkinson's disease. There's a lot of work done in Oxford by Michelle Hugh and co that shows that these are almost for want of a better word, the sort of precursor to Parkinson's, and so
sort of prodromal phase of the
prodromal,
disease.
I suppose. But it's there's a lot of non-motor features that seem to contribute, but the gut is one of those that, you know, whilst it does contain the neurons, is probably not readily associated with the neuronal degeneration, and so maybe it gets left out a bit. But there is a lot of work going on in it, and it probably ought not to be. And I think that there is definite. It might not be directly linked to the things we're eating that's causing it, because there might well be some underlying genetic differences that cause them to be more susceptible to change. But if you think about it, it's another highly energy dependent area of the body. So,
and we know that if you have issues in the gut, you can get a leaky gut, and then things can leak out into other systems that can cause an inflammatory response in your body, can cause an inflammatory response in the brain, and that's never good if it becomes a chronic thing. So I guess it's one of those things where if you have a trigger, then it can just sort of cause a cascade of problems down the line. I find it's really fascinating, the links between the gut and the brain, because I think historically we think of these as very separate things. But actually I think in the last decade we've really started to realise how influential the brain is on our gut and the gut is on our brain, which is, I think, it's a really exciting area of research. And so just to kind of think more about where the future Parkinson's research is going, what do sort of future therapeutics look like? There was a recent story that I just thought was amazing, which was about a woman who says that she smelt her husband's Parkinson's disease before he was diagnosed. And she basically just thought he smelled different and musty, and she thought it was just him. But then when he was diagnosed and she went along to a Parkinson's group, she realised that the whole room smelled like he did. And I would just love to know what you guys take is on that, and where are therapeutics heading in Parkinson's disease?
I think that story is awesome. It's so
amazing.
Fascinating. Perdita Barron up in Manchester is really heavily involved in trying to transfer that. Because, yes, she's just a super smeller, I suppose. She can smell and detect, and she can even get down to the level of maybe stage of Parkinson's as well, which is just, incredible. Oh, yeah, she's amazing. And so they're trying to get towards a way of then sort of quantifying that and understanding what's being expressed in the sweat of these individuals, because effectively, if you could then just mop somebody's brow and then analyze it and be like, you're early stage, we're now getting into the realm of, you know, biomarkers that can tell us about earlier and earlier stages of Parkinson's disease. And that's going to be really critical in treating, right? Because the earlier we can treat, the more chance we have of staving off the onset of
Absolutely.
the cell death. And it all full circle. It all comes back down to trying to stop the dysfunction in those at the synaptic level. So if we can get that to that stage, then we can be like, well, we can give you a drug to stop the breakdown, or at least delay the breakdown of the neurons, which means that you'd end up with developing symptoms much later in life, like, you know, instead of 60,
Amazing.
70, you're doing it.
I'm
80,
so
90.
glad that, because she must have thought she was like, what is going on here? I'm so glad that she kept talking about it, because she's clearly kept saying, I can smell something, and eventually it's you know, people have listened to her and said,
I wonder how many other people had done that before and kind of gone, I'm not going to say anything because that's just rude, or I don't know what I'm talking. Do you know what I mean? You all smell different. That is a bit of a.
That's a really good point. I don't know. I think she's amazing. Sarah, what do you think about therapeutics and Parkinson's and where are we headed?
I feel like I don't have good answers for you, because my approach to the disease is sort of very basic, very fundamental, foundational, going back to what I think are the earliest causes of these diseases. So starting from the environmental exposure,
your strategy would be prevention, basically. So, how do we prevent?
I think there's a good argument for that, like Dayne already said. The earlier you can get in there, the better. So twofold ways. One, if you've had a lot of neurodegeneration, I don't see how we can recreate or rebuild the lost neurons, the lost connections and everything. I don't think that's realistic. So we need to stop it as soon as possible. There's maybe even an argument that it is a cascade. So if you don't get in early enough, it's basically unstoppable. That might be an option too. And in either of those cases, we need to get in there as early as possible, so that the damage isn't even done. So I think that is absolutely right. But the other reason why I go to the environmental and the genetic risk factors is because I really want things that are mechanistically causal to the disease, right? So we have lots of associations of different things with diseases, and Dayne mentioned, for example, sleep disorder, REM sleep disorder, and different things that's been associated with various types of dementias and neurodegenerative diseases. But the causality is really not that clear. So is the sleep disturbance, is it increasing your risk for the disease? And there is some evidence for that, because if you don't sleep well, clearance of aggregates in your brain, things like the Lewy bodies that we talked about, or other aggregates in other diseases, doesn't work as well. So it's certainly not good if you don't sleep well. But equally, people who have these diseases tend to not sleep so well. So the sleeping poorly might be a consequence of the disease and of ongoing neurodegeneration, and it's just a vicious circle that is driving each other. So it's hard to establish causalities for many of these factors, and that's why I sort of want to go back and I really want to understand what is causing it, because if we can act on the pathways and the mechanisms that are the original cause and onset of the disease, then we have the best chance of preventing and stopping it.
So Sarah, do you want to work on both? Do you want to work on both the sort of epigenetics and the genetics? Because you need to look at the genetics to see the initiation and what that's actually driving. Absolutely. And then the epigenetics and how that model. Yeah, because that's fascinating. Your stuff is awesome.
I'm maybe unusual in that I don't see epigenetics as separate from genetics. And I know genetic variants can drive epigenetic mechanisms. And not everyone would agree with me. Some people tend to think of epigenetics as something that is very malleable and environmentally driven. But I don't think that's right. I use sort of more biological definition, like biochemical mechanisms that regulate gene expression, basically. And so I really want to understand it, particularly in the context of the genetic risk and background. So yes, I'm really keen to combine these two things together and leverage the causal information that we have from the genome, because your genome is basically set at birth, and it's not going to be changed by any influences, environments, and anything. So therefore, we can leverage it to understand what is a cause and what is a consequence.
We should work on the iPSC RBD lines that we have, because not to lament the sleep disorder.
Collaboration alert!
Yeah, let's do it. I think we should do it. Because, no, for sure, because I think it's fascinating. Yeah.
Right.
It's true. And the sleep disorders, when talking to people with Parkinson's, they actually say that, well, some of them, not all of them, talk about how the non-motor features are actually much more impactful on their daily life. I mean, anxiety. If you have a sort of a wave of chronic anxiety, that's going to be quite debilitating, right? And sleep. I don't know about you guys. If I don't get good enough sleep, which I often don't, my anxiety goes up. The synaptic connections don't work as well. So cognitive processing and problem solving becomes harder. When you're talking about sort of a cyclic event, Sarah, I truly believe that that's exactly what happens. So if you imagine every time you don't sleep properly, your coordination goes. So movement becomes harder, your processing becomes harder, your anxiety levels do go up. And these are all symptoms that happen in Parkinson's disease, and they seem to be sort of driven. So maybe it's initiated by Parkinson's disease, but I think the sleep may well make it worse. And so it's one of the things that actually we really want to work on in the lab, because we think it's probably turning up quite early, and it's something that maybe if we could modify, may delay the perpetuation of the symptoms.
Super interesting. But also, dear, I slept very poorly last night, and these are not things.
No, these are not great. But I mean, I suppose it's then how you. Let's not too doom and gloom. I don't think you're causing yourself to have PD just by not sleeping properly. But
you've got the weekend to catch up.
It's true.
This is what I say about people with their diets as well. Is that I think we sometimes focus too much on like a single day and what did I eat that day. And it's the same with sleep, like did I sleep well that day? It's better to look at it over a week, because if you have 1 out of 7 days of not eating well, if you've eaten well 6 of the other days, then that's really good. And I think it's the same maybe with sleep as well. Okay, you might not have slept well for 1 day, but if you've slept well the other 6 days, like because people get very anxious about their lifestyles, and that's not going to help it. That's not going to help you have a good lifestyle if you're anxious about your lifestyle. So
exactly, but it is Friday, and I am going out, and so sleep is going to be great.
True. Just have a long lie-in tomorrow. That'll be fine, right?
Absolutely. Well, thank you both so much for coming and speaking about Parkinson's disease today. It's been absolutely fascinating. And I think I'm so excited for your new collaboration that's going to happen.
Yay! It's going to be awesome.
Yes, that sounds fantastic. And you both do incredible work, and it sounds like a really exciting field at the moment. So thank you so much for coming to speak.
No, thank you.
Thank you for having us. It's always a pleasure.
Yeah.
I'm afraid that's all we have time for today. However, if you just can't get enough of this topic, then please visit the Dementia Researcher website, where there are several blogs and articles on Parkinson's disease. I would like to thank our incredible guests, Dr. Sarah Marzi and Dr. Dayne Beccano-Kelly. I'm Fiona McLean, and you've been listening to the Dementia Researcher podcast. Bye!
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 Brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK and Alzheimer's Society, supporting early career dementia researchers across the world.